Good afternoon, everybody. It is my great pleasure to invite Dr. Ray Chang to our physiology seminar. I have actually personally known Ray for a long time because we were in the same college class in the Department of Biology at Tsinghua University. Ray impressed me a lot back then because he liked to choose advanced mathematical courses that most biology students would shy away from. And he did really well in them. I have a feeling that his strong analytical thinking has really facilitated his research, as you will see from his presentation. So after graduating from Tsinghua University, Ray did his Ph.D. in neuroscience with Emily Lehman at the University of Southern California, where he studied the molecular mechanism of taste transduction. So he discovered a proton channel that is critical for sour taste sensation. He then did his postdoc with Steve Liberlis at Cell Biology Department, Harvard Medical School. So Steve has been doing some pioneering work in deciphering the molecular and cellular mechanism of the internal vagal sensory system. In Steve's lab, Ray identified a number of sensory neuron types in the vagus nerve that play important roles in respiratory and GI physiology. In 2018, he was jointly recruited by the Department of Neuroscience and Cellular and Molecular Physiology at Yale University School of Medicine as an assistant professor. In his own lab, Ray is trying to develop and employ state-of-the-art molecular, genetic, and imaging approaches, such as single-cell-seq and RNA-scope, to better understand the diverse body-to-brain interfaces and to develop novel neuronal-based therapeutic strategies for disease intervention. And Ray received the 2019 NIH Director's New Innovator Award, also known as DP2, and the 2021 McKnight Neurobiology of Brain Disorders Award. Without further ado, I would like Ray to take over and start his presentation. Thank you, Ray.
Okay, so first I want to thank Xing for the very nice introduction and for his invitation. It's really my pleasure to have this great opportunity to talk about my research in front of this wonderful department. My lab is generally interested in the interactions between the visual organs and the brain, or the nervous system. And today I'm going to share with you some of our recent progress on the molecular and functional dissection of the vagal interoceptive system. So we all know that the ability to detect the changes around us is critical in our daily life. Therefore, there are a number of sensory systems designated to detect these changes in our external environment. For example, we can see, we can hear, we can smell, we can taste, we can feel the gentle touch and pain and temperature changes through the somatosensory system. But on the other hand, it's equally important to detect the changes that happen inside our bodies. And this ability to detect these changes in organ status is called interoception. And we actually have a wonderful interoceptive system that can constantly and precisely monitor the changes happen inside our bodies. Just to give you a few examples, that we can detect every single heartbeat and provide a beat-to-beat regulation of the cardiovascular functions. We constantly monitor the inhaled air contents within the lung and airways. And we all experience that when we inhale some irritants, we'll make a defensive responsive response, which is cough, to try to get rid of these inhaled harmful gases as soon as possible from our body. There are extensive sensory fibers running along the gastrointestinal tract. And these sensory neurons are trying to detect ingested nutrients or toxins, and they help to coordinate the the motility of the GI tract and the digestive processes, and also control the feeding behaviors. Our body also releases a number of chemicals, including hormones and neuroinflammatory cues. And we know that the neuroimmune interactions is now a very hot topic, and we are only at the beginning of understanding this area. So my lab is generally interested in how these internal organ changes are being detected by the nervous system. And one of the focus we are working on is the vagus nerve. And I hope you can appreciate why we are interested in this system by just looking at this image. So the vagus nerve is the 10th cranial nerve. Its name vagus came from Latin. It means wanderer. So you can see from this image that this nerve has this wandering trajectory that provides extensive innervations to a large number of organs in the neck, in the chest, in the abdomen, including the lung and airways, the heart and vessels, the larynx, pharynx, and the esophagus, stomach, duodenum, small intestine, large intestine. I mean, almost most of the GI tract, as well as the liver, the kidney, the pancreas, the immune organs. So the vagus nerve is very well known as a major component of the parasympathetic nervous system that uses acetylcholine as neurotransmitter to directly control body organ functions. But it's actually much more complicated than that. It's a mixed nerve. It contains not only those motor efferent neurons, but also the sensory afferent neurons that send their projections to most of the organs I just mentioned, where they will detect these organ changes and collect these sensory information all the way up to the brain. The cell bodies of these sensory neurons are located in a pair of vagal ganglia called the nodose and jugular ganglia, which are located right outside of the skull. And centrally, these sensory neurons project into this very specific brainstem region called the nucleus of solitary tract, or the NTS. Unlike the motor neurons, which uses acetylcholine as a neurotransmitter, these sensory neurons use glutamate as neurotransmitter. Therefore, we can easily separate them and target them individually using modern mouse genetic tools. So as shown in this image, the red represents sensory neurons and sensory fibers labeled in VGU2 iris cray with a cray-dependent T-tomato reporter. So VGU2 is a glutamate transporter that's expressing every single vagal sensory neurons, but largely absent from vagal motor neurons. On the other hand, the vagal efferent fibers are labeled in green using the Chat GFP animal. So you can clearly see that the red and green fibers are traveling together in the vagus nerve bundle. And if you take a transactional view, you can even see that majority of the vagus nerve, about 80% of the vagus nerve, is composed by sensory fibers that is labeled in red. So as I mentioned earlier, these sensory neurons can detect a large variety of sensory cues all over the body, including the airway, the air contents within the lung and airways, all the pressure changes within the lung and airways, all the pressure fluctuations associated with every single heart contraction from the heart and vessels, all the ingested food contents, and inflammatory cues. So there's no question that the vagus nerve represents a major interoceptive system. But our question is, here I want to mention that in mouse, there are two vagus nerves. There's left and right. And together, there are roughly 5,000 sensory neurons, which are responsible for presenting so many different sensory cues from different visceral organs to the brain. So our question is, how is this vagal interoceptive system organized so that it can precisely present these numerous different organ signals some of them, many of them are even physically distinct. How does the system present these signals precisely to the brain to appropriately regulate corresponding physiological functions? And years ago, when I was in Steve Libera's lab, we came to a hypothesis that there might be different sensory neurons in the vagus nerve that each will target a specific organ, where they will detect a particular stimulus from that organ and control a selective physiology. And to test that hypothesis, we have generated a number of genetic tools to interrogate the anatomical feature, the response properties and physiological functions of genetically defined vagal sensory neuron populations. For example, we use optogenetics in the vagus nerve to selectively activate targeted vagal sensory neuron populations. As example, we showing you VGU2 iris cray here, which labels all the sensory neurons but not the motor neurons. When we introduce a Chang rhodopsin reporter into these VGU2 positive neurons using the VGU2 iris cray animals, in these anesthetized animals, when we expose the vagus nerve, we can place a optic fiber right on top of the nerve for light activation, light delivery. And you can see on the right, this is a real image from a mouse that the yellow arrow indicates this is the vagus nerve. And here, this is the optic fiber, which is 200 micron in diameter. And you can see the size of the vagus nerve in the mouse. And upon light stimulation, you can see that the vagus nerve can be very well illuminated while the surrounding tissue will not. And in this animal, if we actually record nerve activity using a pair of electrode, you can see that upon light stimulation, we observe very robust neuronal activations. And this activation is different from conventional electrical stimulation in that this is specific effects from only the sensory neurons in the vagus nerve. So you may wonder what will happen when we activate these sensory neurons in these animals. And here I'm going to give you one example of the optogenetic control of breathing. So this is the VGU2 iris cray crossed with cray-dependent Chang rhodopsin animal anesthetized on the table. And the vagus nerve is gently exposed where at this angle you cannot see it. And this is the optic fiber that's pointing right on top of the nerve. And hopefully you can pay attention to their chest movements to get a sense of their breathing patterns. So before light stimulation, the animal has a very stable breathing pattern. And during light stimulation, you can see the animal just basically stop breathing right away. And after light stimulation, the animal starts to breathe again, which clearly demonstrates that these sensory neurons have a very potent role in controlling of respiratory functions, in particular the breathing. So using a similar approach, we identified two sensory neuron types that differentially control breathing. So activating one neuron populations that express P2R1 as a marker will potently inhibit respiration. And this neuron population only represents roughly 10% of total sensory neuron populations in the vagus nerve. On the other hand, when we activate a different neuron population, the non-overlapping population, we saw this rapid and shallow breathing in these animals, suggesting this is a defensive pulmonary responses. Again, this shows the power of using genetic tools to functionally dissect the physiological roles of physically intermingled but genetically distinct neurons in the vagus nerve. So another tool that we have developed is in vivo calcium imaging using a genetically encoded calcium sensor GCAM to study the neuronal activities in the vagus nerve, like how they respond to different organ signals or organ changes. So the idea is this is animal express GCAM in the vagal ganglion, And in these anesthetized animals, the ganglion can be gently dissected and placed onto a stabilized imaging chamber and imaged using a confocal microscope. Here you can see that this is a vagal ganglion with GCAN3 signal. And we can see very robust neuronal activation in response to this positive stimulus, which is electrical stimulation, suggesting that this tool is able to tell us the neuronal activities in real time of these vagal sensory neurons. And using this approach, we identify two sensory neuron types that differentially sense gut signals. So one neuron population that express GLP-1R, so most of the stomach stretch responsive sensory neurons in the vagus nerve will express GLP-1R, as shown in this GLP-1R cross with T tomato reporter mice. So one advantage of this in vivo imaging tool is that we will be able to record the neuronal activities from hundreds of neurons simultaneously at a single neuron resolution. And more than that, we will be able to tell what neurons they are when combined with a Cray-dependent, Cray-assistant neural identification approach. For example, in these animals, we'll cross in a GLP-1R Cray and a Cray-dependent T tomato reporter. And then we can determine that most of these stomach stretch neurons are GLP-1R positive. On the other hand, we also saw that most of the neurons that respond to intestinal nutrients, about 60% of the neurons that respond to intestinal nutrients express this GPR65 as a neuron marker, and these neurons do not respond. The GPR65 neurons do not respond to stomach stretch. So together, these results suggest that there might be genetically definable vagal neuron populations with different response properties and physiological functions in the system, which supports our hypothesis. In addition to that, we find something really interesting, that when we, in the same animal, when we deliver multiple different stimuli, including stomach stretch, intestinal perfusion of nutrients, and lung inflation, you can clearly see that there are different cohorts of vagal sensory neurons that respond to different stimuli given to different organ systems here in this particular case, suggesting that there might be parallel strains in the sensory vagus nerve that present different physiological signals from different organs to the brain. So our major question here is, how is this vagal interoceptive system organized to present numerous body signals to the brain? And the first question we want to ask is, how does the vagus nerve discriminate signals from different organs? I mean, within the body, the vagus nerve is not the unique nerve that innervates multiple organs. And similarly, there are other peripheral nervous systems that also innervate multiple organs. For example, the DRGs, the dorsal root ganglion cells, sensory neurons, also innervate the skin and many of the internal organs, as well as the sympathetic neurons. So in terms of to ask, how does the organs represented in these peripheral nervous system? So we look at the DRGs and we look at the sympathetic neurons, and it seems like there is a topographic organization within these systems. So both the DRG neurons and the sympathetic neurons in the sympathetic chain are organized into different ganglia that's grouped along the rostrocaudal axis of the body. And according to their positions along this chain, they innervate different segments of the body within the neck, the chest, abdomen. So there is a topographic organization there. When we think about the vagus nerve, there's only one ganglion per side. I mean, there's a left and there's a right. So we want to ask whether it's also possible that the different organs within the neck, chest, and abdomen topographically organized within the vagal ganglion. You know, some regions of the ganglion designated for neurons innervate the lung, and some regions of the ganglion are designated for neurons that innervate the heart, for example. So to answer that question, we did a retrograde tracing to look at the organization of neurons that innervate different organs within the vagal ganglion. So here we use retrograde AAVs, also called AAV-RGs. So we injected different AAV-RGs encoding different reporters into different organs. For example, here we injected AAV-T tomato into the heart and AAV-GFP into the stomach. And then we're looking at the vagal ganglion to see how the cells are distributed within the ganglion. And now we can see that there are T tomato positive cells that represent the ones that innervate the heart, retrograde labeled from the heart, and there are GFP labeled neurons that are retrograde labeled from the stomach. And one thing, and so we also look at the heart versus esophagus, heart versus intestine, and many other combinations that I'm not showing you here. So one thing we notice is that largely these neurons that innervate different organs are spatially segregated. In particular, when these two organs are far away from each other or not functional related to each other. So they're spatially segregated within the ganglion. However, if there is a topographic organization of these neurons within the ganglion, for example, like one part of the ganglion is designated for the heart neurons, one for the stomach neurons, then we are expecting to see when we measure the distance between the neurons to the center of the ganglion, all between the neurons that's labeled either from the same organ or from a different organ, we are expecting to see a big difference if the neurons that innervate different organs are indeed topographically organized within the ganglion. And what we observe is it seems like the neurons that retrograde labeled from different organs or different organ pairs are organized in a salt and pepper pattern, meaning that they're equally and randomly distributed within the ganglion. So this data suggests that there is not, we have not observed a topographic organization of visceral organs within the vagal ganglion. So if the organs are not encoded in a spatial organization, is it possible that this critical information is encoded by genetics? I mean, we know that even the same sensory inputs, like stretch of the vessels and stretch of the stomach, although they have the same sensory modalities, but because they're at different locations or different organs, they're going to trigger very specific physiological functions. So we do think this organ information could be really important to determine the physiological function of organ signal and should be preserved or encoded in the system. So to test whether this information is encoded by genetics, we first want to know the molecular architecture of these sensory neurons in the vagus nerve. So we take out the neurons, the cell bodies of neurons located in the vagal ganglion, nodose and jugular ganglion, which is labeled here, and then we did single cell dissociation, and then we did enrichment for neurons to remove most of the other glial cells. So as shown here, you can see that we can isolate individual single neurons. And then we did single cell RNA-seq using the 10x genomics-based platform and analyze the data using thread package. So from each run, we can largely analyze roughly around 5,000 neurons with a pretty reasonable number of genes per cell and a pretty decent sequencing depth. So together, we analyzed 13,000 vagal sensory neurons from 40 gender and age matched wild-type animals. And these 13,000 neurons clustered, unbiasedly clustered into 12 large subpopulations of 52 small subclusters. So the large populations are labeled with the letters from A to L, with different color, and the smaller subclusters within each subpopulation is given by a number, for example, A1, A2, A3, A4. So first, we want to know if this single-cell RNA-seq data can faithfully represent neuronal clusters of gene expressions in the sensory vagus nerve. So we look at the expression of many known genetic markers here. So we know that TRPV1 is a marker for most of the slow conducting C fibers, and piezo2 is a marker for many of the fast conducting A fibers, and GPR65 represents a small fraction, but TRPV1 negative C fibers. And these three genes from our previous studies are shown that they are expressed in different neuron populations. And you can see that indeed TRPV1 labeled in red, piezo2 labeled in green, and GPR65 labeled in blue. They are expressed largely in different non-overlapping neuron populations in the single-cell RNA-seq. And also, we can identify, we tested the markers that previously identified, including GABRA1, TRPA1, TMC3, UTS2B. They're all expressed in the predicted neuron populations. And our single-cell RNA-seq data is very consistent with recently published single-cell database in the vagal sensory neurons, further confirm that the reliability of these data set. Then we were able to identify genetic markers for individual vagal sensory neuron subpopulations. As shown in the dot plot, you can see that each neuron subpopulation is marked by 2 or 3, can be defined by 2 or 3 marker genes, and together these genes will, in a combinatorial way, determine the precise molecular identity of these vagal sensory neurons. Then we want to see whether we can confirm this and really determine the neuronal identity of neurons using spatial transcriptomics. So we performed RNA-scope high-plex assay for 24 marker genes that listed, most of them are listed here on the dot plot, that can in a combinatorial fashion can define the precise neuronal populations. Here is a video to show you that on the single vagal ganglion cross sections, we can determine their expression for a particular sensory neuron, the expression of 24 genes, marker genes. And using this approach, first, I want to show you that, because it's 24 genes, it's really hard to see them. So I picked some genes to validate the single-cell RNA-seq data. So you can see that the red labeled genes, TMC3 and TRPA1, are expressed in a large neuron populations, but non-overlapping neuron populations. And this is exactly what we saw when we look at the TRPA1 and TMC3 labeled neurons in RNA-scope. On the other hand, the RUNX3, GABRA1, GM765, UTS2B, the blue labeled neurons, they are 4 genes that marks 4 individual smaller neuron populations. And you can see that when we put these genes together, you can see they each mark an individual neuron cluster. So together with these 24 genes, we can pretty well define the genetic identity of a sensory neuron on the slide. And when we look at the percentage of the neurons within each subpopulations, you can see that the numbers determined using RNA-scope and the single-cell RNA-seq are pretty consistent, suggesting that not only our single-cell RNA-seq data set is reliable, but also we are able to determine the vagal neuron genetic identity on the slide using this spatial transcriptomics. But also, when we look at the single cell RNA-seq data, it is really hard, or it's almost impossible for us to determine, which cluster represents the neurons innervate which organ, right? It's hard to determine whether cluster A innervate the heart or cluster B innervate the lung, or even if that's true in this case. So to address that, we further developed, we want to label these neurons that project to different organs and analyze them, their genetic features. So we developed a tool called the single neuron analysis based on projections. We call it SnapSeq. The idea is, unlike the traditional neuron tracers like the DIE and CTB or the neuron retrograde beads, this retrograde AAVs will provide genetic handle to those labeled neurons, and thus is ideal for this multiplex sequencing. So we engineered the AAV, retrograde AAV expressing a T tomato reporter, and then we inserted a unique projection barcodes into this vector of this T tomato retrograde AAV vector. And right before the poly A tail, because this, the single cell RNA-seq based on the text genomics, B3 is highly 3' bias. So the position of the barcode will facilitate the detection of the barcode. And then we generate a series of SnapSeq comparable AAVs, we call the SnapSeq AAVs. And then we injected each organ with a different AAVs with the different barcodes. So together we injected 7 major organs within the same animal, including the lung, the heart, esophagus, stomach, duodenum, colon, and pancreas. And then so in the same animal, the sensory neurons will be retrogradely labeled simultaneously from all these 7 organs, but each neuron labeled from an individual organ will be labeled with different barcodes. And then we'll take out the sensory neuron from these injected animals and isolate them and sequence them. And hopefully we can find the barcodes and then find the neurons based on their barcodes and study their genetic features. So before I get to that, there are a number of control experiments we need to confirm. So first, we want to know if the insertion of this barcode will disrupt the infection efficiency. So in the upper image I'm showing you, this is a vagal ganglion in an animal with stomach injection of a regular AAV GFP. And the bottom image shows that this is an animal with stomach injection of AAV, with SNAP AAVs with one of the barcodes. You can see that the infection efficiency is comparable between the regular AAV, retrograde AAVs and the SnapSeq AAVs. On the right, I'm showing you that we were able to detect these introduced barcodes using from the vagal neurons using RT-PCR. So the red color represents an animal with stomach injection of SnapSeq AAV with with the barcode 2, like UPV2, and then the bright red indicates then we did the RT-PCR using a primer that recognized UPV2 in these ganglion, and you can see we saw a bright band here. On the other hand, when we use a primer set that recognized a different barcode, we did not see that band. And in the animals that we did not receive any barcode injections, we did not see that band either. Indicates that in these injected animals introduced with different barcodes, we were able to detect these barcodes from the sensory neurons. So next, we sequence these neurons and look at what we see from the single cell analysis. So first, we compared the UMAP plot from the SnapSeq studies with the control studies, and you can see that after the SnapSeq approach, like with the injection of the organs, the neurons look exactly the same as the ones we got from the control experiments, suggesting that largely this SnapSeq approach did not change the cell clustering using single cell RNA-seq. And also we count the percentage of neurons within each large vagal neuron subpopulations or each small vagal neuron subclusters. And you can see that the percentage of neurons in each population is very consistent between the control RNA-seq and the SNP-seq. We further look at the expression patterns for many marker genes, including the ones listed here, and you can also see that the gene expressions between the SNP-seq and the control RNA-seq data is very consistent. So together, this data suggests that with the SNP-seq approach, the sequencing result can still reliably represent the sensory neuron clusters populations as well as the gene expressions in the vagus nerve. So next, we want to ask whether we are able to identify those neurons that innervate different organs using the barcodes. And the answer is yes. So here I'm showing you the UMAP plot colored by the barcodes that from different organs. It's probably not very easy to get an idea from this image, but the bottom line is we were able to identify unique gene markers that are highly expressed or highly preferentially expressed in the neurons that innervate different organ systems, suggesting that indeed it's possible that these the neurons that project to different organs are genetically encoded. Moreover, so we look at the neurons that project to, first we look at the neurons that project to different physiological systems. So including the heart as representing the cardiovascular system, the lung representing the respiratory system, and the esophagus and many gut organs, GI organs represent the gastrointestinal system. So it's very clear that the lung labeled cells genetically segregated from the neurons that label from the gut or the heart. From this 2D UMAP plot, it's a little bit hard to tell whether the heart and gut cells are intermingled together, but we find that when we look at the 3D UMAP, it's very clear that the heart labeled neurons are clustered together in a small branch that deviates from this major plane labeled by the gut cells. And you can have a better idea from looking at the 3D video of the 3D UMAP plot that the red labeled heart projecting neurons spatially genetically segregated from the blue labeled lung label, the blue color lung labeled sensory neurons and the green color gut labeled sensory neurons. And in addition, we were able to find transcription factors that are differentially expressed in the neurons that innervate the lung, the heart, or the gut. And many of those transcription factors have been reported in the DRG system that are guiding the neuronal projections. For example, the POU4F1 that we that preferentially expressed in the neurons that project to lung also were reported to have a similar role in guiding the DRG neuron projections. We then ask if these sensory neurons that project to different organs are using a similar approach or similar ligand receptor pairs to interact with organ cells, or they're using specialized ligand receptor pairs to interact with cells within different organs. So so for that purpose, we perform the cell phone DB analysis, which is a cell-cell interaction analysis to predict the potential ligand receptor interactions between groups of neurons. And interestingly, that we saw that the heart neurons make a lot of interactions, predicted interactions with different cardiac cells versus the lungs innervating sensory neurons also express a lot of potential receptor ligand pairs interacting with the different cell types within the lung. And interestingly, that these ligand receptor pairs between the sensory neurons innervate different organs and the organ cells are different, suggesting that these sensory neurons use different receptor ligand pairs to interact with different organ cells. We next look at the organs within the GI tract. As these organs share many functional similarities, when we look at their distributions on the UMAP plot, it's very clear that these neurons are not segregated as clearly segregated as the ones among the lung, heart, and gut. So they're kind of a little bit intermingled together. To better understand what is the organization for different GI organs in the sensory neurons, we take out all the neurons that highly innervate the GI organs, and then we look at if they are equally innervating all the different GI organs, or they have a preference for their targets along the GI tract. And interestingly, what we saw is each of these, most of if not all of these GI innervating sensory neuron populations will have a preferred target regions along the GI tract. For example, these neuron clusters will preferentially innervate the esophagus versus these ones will preferentially innervate the colon. Right? And to better quantify this result, so we gave an organ index for the GI regions or GI organs along this GI tract. So for example, esophagus, we get 0, the esophageal sphincter, we get 1, stomach, we get 2, pyloric sphincter 3, duodenum 4, and transverse colon, we get 5 based on their locations along this GI tract. And then we ask for each of these gut innervating sensory neuron populations, what is their organ index, meaning that for these neuron clusters, because they're highly prefer the esophagus, they will get an organ index very close to 0. And for these ones that highly preferred for colon, they will get an index close to 5. And for these ones that target preferentially along a middle region along clusters on the UMAP plot. And surprisingly, we saw this trajectory that seems to be able to predict the target locations of these neurons, sensory neurons along the GI tract. For example, here at one end of the trajectory, these neurons will have a very low index score, meaning that they preferentially target the esophagus, versus at the other end of the trajectory, these neurons will have a pretty high organ score and they were preferentially targeting the colon. And to better assess this relationship between their positions on this GI organ trajectory and their periphery targets along the GI tract, we measured the relative distance of each GI organ along the GI tract. For example, from the start point of the GI tract, which is the throat, all the way to the end of the GI tract, which is the rectum. So we measure the relative distance of each organ along this GI tract. And this number is plotted in the x-axis. On the y-axis, we plotted the number of each sensory neurons, the projection numbers of each sensory neurons onto this GI organ trajectory. And clearly you can see there is a very linear relationship between this, their projections onto this GI organ trajectory and the positions of the GI, the organs along the GI tract, suggesting that their periphery targets along the GI tract can be very well predicted based on this organ trajectory on the UMAP plot. So together, this data suggests that instead of a topographic organization of the organs in the sensory vagal neurons, there is a genetic organization of the periphery organs within the sensory vagus nervous system. So in addition to this GI organ trajectory, if you remember, is in this direction. We also identified a second trajectory on the UMAP plot using slingshot, which is a software commonly used to predict cell projections from these single cell studies. And surprisingly, we found this second trajectory is kind of in orthogonal orientation to the first organ trajectory we identified. So we wonder if, similar to the first organ trajectories, if the second trajectory can also encode some useful information in the nervous, in the sensory vagal neurons. So we identified using the trait-seq the genes differentially expressed along this trajectory. For example, GPR65 and SST are highly expressed at one end of the trajectory. TRPV1 and DRD2 kind of label neurons that sits in the middle part of the trajectory, and AGTR1A label neurons that sits on the other side of the trajectory. I mean, along with many other markers expressed along this trajectory, including many transcription factors, as well as molecules involved in neural development and axonal projections. So next we want to ask, if this trajectory is meaningful, is it possible it's also encoding some information? And therefore we take advantage of mouse genetic tools and label these neurons in according query lines to look at if they have some special features that can fit along this trajectory. So for that purpose, we did virus-guided anatomical tracing by injecting a query-dependent T tomato virus into the vagal ganglion in the corresponding GPR65, SST, TRPV1, DRD2, and AGTR1A query animals to specifically label these neurons. And then after a couple of weeks, we'll take out the organs and then we perform tissue clearing to so this is the stomach from these injected animals, and then after clearing, you can see that the stomach gets pretty transparent. And in this way, we were able to use light sheet microscopy or confocal microscopy to get a high resolution image of the projections from these fluorescent labeled sensory fibers. And what we observed is quite interesting. I'm just here showing you a couple of examples. So for GPR65 and SST, we observed that these neurons will project to the mucosal layer of the GI tract, GI organs. So SST is mainly limited in the stomach, but for GPR65, it's observed all the way from esophagus to stomach to intestine, and all the majority of GPR65 terminals observed in this mucosal layer, which is the most inner layer of the organ, of the GI organs. Sorry, I forget the label here. So this is from the DRD2. So when we look at the DRD2 terminals observed, again, along most of the GI organs we observed, including the stomach, esophagus, duodenum, and colon, as well as the heart. And majority of DRD2 positive fibers are located within the muscular layer, which is kind of between the surface and the inner mucosal layer. So these fibers are forming terminals within the muscular layer. On the other hand, the AGTR1A positive fibers were also observed along all the GI organs as well as the heart. And these neurons form terminals in the connective tissue layer, which is even in the outer layer of the organs. So within the GI organs, these AGTR1A positive fibers form IGLEs within the mesenteric layer. And within the heart, these AGTR1A fibers form flower spray-like terminal structures on the epicardial layer of the heart. And a common feature of these environment is that they are all very elaborative, highly connective tissue containing layers. So we feel like if we gave the tissue layer a score, and the outer connective tissue layer we give a score of 2, the muscular layer, the middle muscular layer we give a 1, and the inner epithelial layer we give a 0, and then we calculate what is the tissue layer index score for these sensory neuron terminals formed from these genes differentially expressed along this trajectory. We saw something very interesting that this tissue index score correlates very well with their positions along this tissue projection trajectory, suggesting that this trajectory may actually encode the tissue layer of the vagal sensory terminals. And again, when we plot these numbers on the UMAP plot, you can clearly see that the tissue score follows this tissue trajectory along this trajectory. And also, when we plot the pseudo depth score versus the tissue layer index, you can see that there is a very well correlated linear relationship between these two, suggesting that this second trajectory is encoding the terminal locations, the tissue layer locations of the sensory neurons. So now when we think about this, we think it makes a lot of sense. So first, the reason is the position, the physiological function of an internal signal is determined not only by which organ is being generated, but also which which tissue layer within that organ is generated. For example, within the GI tract, you can sense nutrient signals or stretch signals, and depends on where the signal is generated, they're going to trigger very different physiological functions. So together, our data suggests that there is a genetic representation of the body's internal space composed by a combination of the organs and the tissue layers in the vagal sensory neurons. So in the sake of time, I'm going to skip this. I'm just going to briefly mention that we also designed a new imaging tool that we can efficiently combine the neuronal activities with their genetic features and their terminal structures together, that together we think there are 3 independent parameters that we identified. The tissue ending types correlates very well with where the tissue layer is. A visceral organ seems to function as the independent parameter, and sensory input is very highly correlated with genetically defined sensory neuron populations. And together, these 3 parameters work as 3 independent dimensions in the vagus nerve, and our data suggests there is a multidimensional architecture of this vagal interoceptive system. And for each of the dimensions, for example, the organs or the tissue layers, the sensory neurons only need to specialize into a few different categories, but in a combinatorial fashion, they will create a large number of parallel streams to present different physiological, different organ signals to the brain. And at the end, I want to acknowledge people that contribute to this project. Most of the work is done by a postdoc, Chang in the lab. And I didn't mention that the calcium imaging, the new calcium imaging tool is performed by a graduate student, Yue in the lab. And also want to thank for the funding sources, and thank you for listening.
Oh, that was a great talk. I think it was a very vivid demonstration of the power of the cutting edge technologies and the insight that could be derived from using those technologies that are otherwise difficult to obtain by conventional approaches. Okay, let's see. Daisuke has a question. Let me. Okay, Daisuke, you can talk now.
Hi, thank you, Shane. Um, I have a question about the sequencing that you did with barcode, that different barcode injected into different organs. Have you seen any class of neuron that project to multiple or project from, I guess, multiple organs? And if so, can you speculate on their function?
It's a good question. So, I mean, that is something we are constantly looking for because there is a hypothesis that one neuron could project to multiple organs and detect, integrate, do a signal integration at the ganglion level. So far what I can say is we observed about 15% of neurons that are double labeled by two barcodes. And more than barcode, there are very, very little neurons that are labeled by more than two barcodes. I think the number is very minimum and can be ignored. So when talking about the neurons that express two barcodes, we noticed that, I didn't show the data here, but we did a correlation analysis, and the two barcodes are mainly from adjacent organs, which is observed when we, for example, like many dual barcode labeled neurons are from esophagus and stomach. And this is also observed when we inject the retrograde viruses into stomach and esophagus. We do see a number of overlapping neurons. And so far, we think the most likely answer for that is because the retrogradely injected virus will spread some distance in the organs, and those neurons that double labeled by two barcodes will likely innervate the junction between those two organs. So to answer your question, we don't see a neuron population that innervate multiple organs and is labeled by multiple barcodes.
Okay, DJ, do you want to ask a question?
Yeah, it's kind of related to that. So in this retrograde labeling experiment, you inject virus, right, in a target organ, and then look at the neuronal signature. And so, I mean, I can imagine that different organs are very different in terms of their accessibility or their size even, right? Say, like if you were to inject into liver versus like say a much smaller tissue like a pancreas. And so how do you make sure that you cover the whole liver, or you only targeting part of the liver, or even which layer of the liver? I don't even know. I mean, so that's the question. Yeah.
That is a great question, and it's interesting you bring up liver because that's the organ we didn't do for exactly the reason that the liver is so big, and it's really hard to know where we injected and where the nerve terminals are. However, for the organs we injected, in particular the heart and along the GI tract, we try to cover the entire volume of the organ that we can. For example, for the heart, we injected the viruses in all the four quadrants of the heart at different depths with a large volume of virus. So so that is my answer, but I also agree with you that, for some regions that, for example, the top surface of the heart or the outer surface of the airways, it is a little bit difficult to target using this approach. For the data, I didn't show that we did a systematic tracing of using multiple cray lines to looking at the neuronal innervation into these 6 or 7 major visceral organs. And indeed, in particular in the lung, we identified that neurons that innervate the airways are not very well labeled by this retrograde injection. But otherwise, in other organs, the anatomical tracing and the SNAP-seq analysis seems to be very consistent with each other.
Yeah, and I probably follow up with a more kind of a more philosophical question here. That is, what is this idea of topographical segregation versus like the genetic, right? So what is the advantage of having a topographical, organization, which you see really in the retina, for example, right? Versus here. So what can you just tell us a little bit what your thinking is about? Yeah.
What I think is, like if there's a topographical organization, it probably indicates that, at the very early developmental stage, these cells will be segregated to get from each other. And then from there, they probably don't need to care much about their genetic features for the organs, right? So they just replicate and proliferate, and that location will be determined for a particular location. For example, the DRGs, they are segregated in the early stage, and then each of the ganglion will be designated to a particular segment of the body. Versus if these neurons are intermingled together, it's more likely to say that they will need some other mechanism to tell where they will go in the future when they actually trying to target to the peripheral organs.
Mm-hmm. Mm-hmm. Yeah, well, we can discuss that later. Thank you very much. Thank you.
Thank you.
So yeah, I have a related question though. So first, since you have observed this almost perfect overlap or congruency between your single cell seq versus the SNAP-seq, do you think you would interpret that as you are not missing any major population that are innervating other organs that you did not inject, such as liver?
Oh, that's a totally different a different question, right? So the SNAP-seq still, we are analyzing all the sensory neurons that we can isolate from the vagus nerve. It's just some of them are given a barcode. So the ones that innervate other organs that's not been labeled, still there.
Okay.
We will not be able to see them if we only look at the neurons that express the barcode, but if we ignore the barcode, they're still there.
Okay, all right.
Okay.
And regarding this topographic versus the genetic coding, do you think the difference, the different genetic profiles you saw across those neuronal populations, are they like intrinsically or pre-programmed as a intrinsic property of those neurons, or they are actually induced after those neurons reach their targets of innervation? Because this distinction would somehow change the interpretation of the data. So what's I'm curious to hear your insight on that.
Yeah, that's a very interesting question. And the short answer is we don't know. I think both possibilities exist. There are data to support that there are these neurons. Some of these neuron clusters show up in a very early developmental stage, suggesting that, and also the data that we use a Cray line to label some of the neurons, and they always form the same terminal structures and detect the same sensory cues, but now the others suggest that these neurons, at least at a pretty early developmental stage, are designated to their terminal structure, terminal locations, and their physiological functions. But on the other hand, the vagal sensory neurons, or in general, the peripheral nerves, can regenerate. So if you cut them, they can project back to their locations. It's not entirely clear whether it's the same neuron can project to its original positions, or but largely as a group, they can project back to their organs layers. So meaning that it's very likely that these mechanisms still persist in adult neurons.
I see. Okay, so Helen has a question. are you able to separate out the nodose versus the jugular cell types like the Force group has done? If so, where do the jugular neurons innervate? And is there a genetic somatopy?
That's a good question. I mean, so far, so for all the data I presented today, that's from the so-called nodose ganglion. I mean, it's actually all the neurons I showed are FOX2B positive neurons. Because in mice, the nodose, jugular, and petrosal ganglia fuse together, so we cannot say for sure they are 100% nodose. There might be a small fraction from FOX2B positive petrosal ganglion, but they are FOX2B positive neurons. On the other hand, the jugular neurons are not included here, and also we observe very little barcodes in those jugular neurons, and the reason is probably because we only focus on most of the organs we focus on is kind of the visceral organs, and it's being reported that the jugular neurons highly innervate the upper airways, which is not the target that we study here.
Okay,
but
I guess.
Right, the bottom line is we are able to separate these very clearly. And
okay.
Yeah.
Daisuke, do you have another question? I saw you raise hand again.
I guess I will just ask a quick question. So are these organ dependent or organ specific pathway completely labeled line in the next ganglia as well, or could some of the pathway be merged? You know, I would assume, or I would think that it may be beneficial to perhaps integrate all the inflammatory signal from all organs, etc.
Excuse me, could you say your first sentence again? I'm kind of a little bit lost there.
Yeah, if you look at the next order connection, is the labeled line per organ completely segregated in the next order as well, or perhaps some modality is integrated? You know, for example, like inflammatory response pathway is pooled across organs.
That's a good question. I didn't show that data here. We did cell type and terminal type specific tracing, meaning that we use a combination of cray lines that target a particular neurons in a particular tissue layer and organ injection of the retrograde tracers to label these neurons and looking at their projections in the brainstem. So far, what I can tell is the organization seems to be dependent on multiple factors. It's not like simply, so first we did not see a better segregated projection using this organ and tissue type specific tracing. Meaning there's a lot of integrations. There are multiple pathways project to the same locations in the brainstem. And second, it seems like their projections is not dependent on a single factor. So it's not dependent on the organs completely. It's not dependent on the tissue layers. It's not dependent on the sensory modalities or the sensory inputs. It seems like all different factors together used to determine its physiological functions and then determine its terminal projections in the brain. So I will say it's definitely not, in their second order, it's definitely not a label line based on organs or based on tissue layers or based on sensory inputs. It might be a label line based on their physiological functions.
Great, thank you.
Sure.
DJ, do you have more questions? I saw your hand is still up. Oh, DJ? Oh,
I don't have question.
Okay, I see. Okay, all right. Okay, I guess that's all the questions. I want to thank Ray again for this wonderful talk and thank people for attending.
Thank you.